Hey guys, welcome to Spec Transfer and to topic 3.6.1.3, control of heart rate from the AQA A-Level Biology specification. As always, let's start with a look at our specification. So we need to know about the myogenic stimulation of the heart and transmission of a subsequent wave of electrical activity, i.e. how an electrical impulse is generated and the path that the electrical impulse takes. We need to know the roles of the sinoatrial node, also known as the SAN, the Atrioventricular Node, also known as the AVN, and Purkinje tissue in the bundle of His. Finally, we should know the roles and locations of chemoreceptors and pressure receptors, and the roles of the autonomic nervous system and effectors in controlling heart rate. So let's make a start. Cardiac, i.e. heart muscle, is myogenic, meaning that the contraction is initiated by the muscle itself without receiving signals from nerves. Here we have a heart diagram and some of the components that we need to know. We have the sinoatrial node, the atrioventricular node, the Purkinje fibers, and the bundle of His fibers, which are a collection of Purkinje fibers. And we'll learn all about these and their various roles in just a moment. So the SAN sets the frequency at which the cardiac muscle cells contract. It sends out regular electrical impulses to the atrial walls, causing the atria to contract. So how does one contraction occur? First of all, the SAN generates an electrical impulse. This impulse then spreads from the SAN across both atria, causing them to contract simultaneously. The septum prevents the impulse from crossing to the ventricles. The impulse then reaches the AVN. After a short delay, the AVN passes on the impulse to the bundle of His, which conducts the impulse down to the Purkinje fibers. Finally, the impulse reaches the Purkinje fibers at the heart apex, which carry the impulse up the walls of the ventricles, causing them to contract simultaneously from bottom up. So why is there a short delay at the AVN? This is to allow the atria to be emptied before the ventricles contract. So how do we actually control heart rate? This is done by the autonomic nervous system. The rate at which the SAN generates impulses is controlled by the medulla oblongata in the brain. First of all, we should know that heart rate is changed in response to internal stimuli. This can be things such as a change in blood pressure, which is detected by baroreceptors, or chemical changes in the blood, which are detected by chemoreceptors. Chemical changes, for example, may be a change in the concentration of carbon dioxide, which would affect the blood pH, or a change in the concentration of oxygen. Both baro and chemoreceptors are located in the aorta and carotid arteries. When one of these receptors detects a change, it sends electrical impulses to the medulla via sensory neurons. The medulla then processes this information and sends impulses to the SAN via either a sympathetic or a parasympathetic neuron. Note that the sympathetic nervous system prepares the body for more activity, whereas the parasympathetic nervous system prepares the body for relaxation. They are antagonistic, meaning that at any given time only one of these is operating. They never work simultaneously. So what determines whether the SAN increases or decreases the heart rate? If an electrical impulse is sent via a parasympathetic neuron, the neurotransmitter acetylcholine is released and binds onto receptors on the SAN. The SAN then sends out electrical impulses at a lower frequency, heart rate decreases, and the body is prepared for relaxation. If the medulla sends electrical impulses via the sympathetic neuron, noradrenaline is released, which binds to receptors on the SAN, and the opposite happens. The SAN sends out electrical impulses at a higher frequency, heart rate increases, and the body is prepared for more activity. So here we have it in summary in a table. If high blood pressure is detected, a high concentration of oxygen or a low concentration of CO2, which would also result in a high pH, then either baro or chemoreceptors, depending on what was detected, send more impulses to the medulla. The medulla then processes the information and sends impulses to the SAN via a parasympathetic neuron. This releases the neurotransmitter acetylcholine, which binds to receptors on the SAN. The SAN sends out electrical impulses at a lower frequency and heart rate decreases. Therefore, the blood pressure decreases, concentration of oxygen decreases, the concentration of CO2 increases, and blood pH decreases. If we have a low blood pressure, a low concentration of oxygen, or a high concentration of CO2 and a low blood pH, baro and/or chemoreceptors send more impulses to the medulla. This processes the information, sends out electrical impulses to the SAN via a sympathetic neuron, and this releases the neurotransmitter noradrenaline, which binds to receptors on the SAN. The SAN sends out electrical impulses at a higher frequency and heart rate increases, meaning that blood pressure increases, the concentration of oxygen increases, the concentration of CO2 decreases, and blood pH increases. Great, that would be this part of the specification covered. We have covered how an electrical impulse is triggered at the SAN and how it then spreads throughout the heart. We have also covered the roles of the sinoatrial node, the atrioventricular node, and Purkinje tissue in the bundle of His. Finally, we have covered the roles and locations of chemoreceptors and pressure receptors, and the roles of the autonomic nervous system and effectors in controlling heart rate. That would be it for now, guys. Thanks for watching. Please subscribe, comment. Next time we will be covering nerve impulses.